Okay, so in this module number 3, we will discuss mRNA and LNP design. So in this lecture, we will introduce RNA lipid nanoparticle therapeutics. So the key idea is that these medicines are essentially systems. The RNA payload determines what biological instruction is delivered, and the lipid nanoparticle part determines whether that instruction survives in the body, reaches the right cells, enters them, and produces the intended effect. Let's start with the first FDA approved siRNA LNP therapeutic. Before we discuss mRNA LNP systems, it's useful to introduce Onpattro, which was the first FDA approved siRNA LNP therapeutic. That was in 2018. In this case, the LNP delivers siRNA to hepatocytes, where it reduces production of transthyretin protein through RNA interference. So what matters for us is this drug demonstrated that lipid nanoparticles can reproducibly deliver RNA payloads to the liver and achieve meaningful clinical effects in humans. That success is what enabled the rapid expansion of RNA LNP technologies that we will focus on throughout this module. Next, let's start with mRNA LNP system. A major milestone for RNA LNP therapeutics came with COVID vaccines. So these were the first mRNA vaccines approved for human use, and they rely on the same core delivery concept we just saw with the siRNA, that is LNP. So here that is encapsulating a chemically modified mRNA payload. In this case, the mRNA encodes a spike protein, and the LNP enables protection of that RNA, and then it delivers to cells, and then sufficient intracellular release to drive protein expression. So what is important for this course is not particularly about this vaccine or this indication, but the validation of RNA LNP therapeutic system at global scale. Now let's start with the three subsystems of all LNP. At a high level, RNA LNP therapeutics can be understood as three interacting subsystems. The first is the payload, the mRNA or siRNA. The payload defines the biological instruction, which protein is expressed, for example, or which gene is silenced. What it does not determine on its own is how efficiently that instruction is delivered in vivo. So the second subsystem is the lipid formulation. So this includes the ionizable lipid, helper phospholipids, cholesterol, and PEG lipids. Together, these components govern RNA encapsulation, particle stability, cellular uptake, and many aspects of tolerability and toxicity. The third subsystem is the nanoparticle that emerges from these components. Typically, a particle on the order of 50 to 100 nanometers. So its size, structure, and surface properties strongly influence biodistribution, circulation time, and likelihood of successful endosomal escape. So a key idea throughout this module is that therapeutic performance is not controlled by any one of these subsystems alone. It emerges from their interaction, and that interaction is what ultimately needs to be understood and later modeled, of course. Let's see, the composition of approved LNP medicines in the next slide. So this table compares the compositions of three clinically approved LNP medicines. Despite the different RNA payloads, the overall architecture is, basically same. All three formulations use an ionizable lipid, a helper phospholipid such as DSPC, cholesterol, and PEG lipid to form the nanoparticle. So what changes across product is the chemistry of ionizable lipid and the PEG lipid, which tunes delivery behavior, tolerability, and biodistribution. Onpattro uses the ionizable lipid MC3. So this was optimized for ApoE-mediated uptake into hepatocytes. However, MC3 was not designed to be rapidly biodegradable and lacks the cleavable ester linkages which used in later ionizable lipids. So these mRNA vaccines use different ionizable lipids, that is ALC0315 and SM102. As I just mentioned, these lipids have ester linkages to promote biodegradability while maintaining efficient mRNA delivery. So what's the key point here? So that successful LNP medicines do not rely on entirely new architectures for each application. Instead, they reuse a common framework and adjust lipid chemistry to balance delivery, efficiency, expression, safety, and manufacturability. A, a critical point in RNA LNP therapeutics is that the final nanoparticle is not defined by just composition. It is shaped by how it is assembled. During formulation, lipids are dissolved in ethanol and they are rapidly mixed with RNA solution, most commonly using microfluidic mixing. So under these conditions, ionizable lipids become protonated and then bind the RNA and nucleate particle formation. When it comes to key manufacturing parameters such as N2P ratio, the mixing method, total flow rate, flow rate ratio, pH. So all these things strongly influence how quickly particles form, how large they grow, and how uniformly RNA is encapsulated. So downstream steps, including buffer exchange, purification, further affect particle stability and surface composition. So the important thing here is that LNP structure is an emergent property of both chemistry and process. So you can see two formulations with identical lipid compositions. They may behave very differently in vivo if they are assembled under different conditions. Here we can see the journey of LNP therapeutic. At each stage, the system operates at a different physical scale, from molecular interactions between lipids and RNA to nanoparticle behavior in solution to cellular uptake, tissue distribution, and finally organism level response. After administration, particles must remain stable in the bloodstream and then, of course, avoid rapid clearance and reach target tissues, right? They then need to be taken up by cells. And then in the cells, they need to escape the endosomal pathway. So RNA gets released into cytosol. And then you see protein expression. So each of these steps introduces new physical constraints. A formulation that works well at one scale can fail completely at another. The key point is that LNP performance is inherently multi-scale. Therapeutic success requires acceptable behavior across all of these levels, not optimization at just one level. Okay, so let's see the key ideas that we learned in this lecture. All approved RNA-LNP medicines share the same three subsystem architecture. First is the payload, maybe siRNA or maybe mRNA, which carries the biological instructions but is chemically fragile and can trigger innate immune responses. Second are the lipid components, an ionizable lipid, um, a helper phospholipid such as DSPC, and cholesterol, and a PEG lipid. So these lipids assemble during manufacturing to form the LNP. Roughly about 50 to 100 nanometer structure that emerges from both the lipid chemistry and controlled mixing. So within that particle, the different lipids play distinct roles. The ionizable lipid enables RNA encapsulation and supports endosomal escape. Helper lipids and cholesterol stabilize the structure and tune membrane properties. And PEG lipid modulates particle assembly and circulation time. At the level of the assembled nanoparticle, this organization shapes biodistribution and dominant cellular uptake pathways. Finally, it is important to recognize that RNA-LNP performance emerges across multiple biological scales, from molecules to particles to cells and then to tissues and ultimately the whole organism. Thank you.